Tracoma, one of the most prevalent eye diseases in the world. Today, this chronic keratoconjunctivitis affects more than 80 million people, 8 million of whom suffer visual impairment. Those most affected live within the gross poverty belt in conditions of misery, with inadequate access to water, healthcare and hygiene. Tracoma remains the world's #1 infectious cause of blindness. What is trachoma? It is a chronic infectious keratoconjunctivitis caused by Chlamydia trachomatis, whose diagnosis is above all clinical. It begins during childhood and may be cured spontaneously or recur and lead to the adult complications of palpebral retractations, which in turn leads to entropion of the upper eyelid and trichiasis that causes progressive and irreversible opacification of the cornea. It is a contagious illness, with flies and soiled linens constituting the principal agents of transmission. The diagnosis of trachomatous conjunctivitis is made by everting the upper eyelid and examining the tarsal conjunctiva with the help of a magnifying glass. Clinically, the classification of Macallan has been replaced by the simplified trachoma grading system, with 5 grades to facilitate the epidemiological study of the disease. Follicular trachoma occurs in the young child and is characterized by the existence of more than 5 translucent or yellowish follicles, at least half a millimeter in diameter. Intense inflammatory trachoma is characterized by at least 5 follicles and very red thickening of the conjunctiva that masks more than half the vessels. Follicular and inflammatory trachoma are also called active trachoma. In the scarring stage, or scarring trachoma, which occurs after repeated infections, lines of scar tissue appear on the conjunctiva. In the entropion trichiasis stage, or trachomatous trichiasis, at least 1 eyelash rubs against the eyeball. Finally, corneal opacities cover the entire pupil. The germ responsible for trachoma is Chlamydia trachomatis, a 300 nanometers microorganism with serotypes A, B, BA and C. Its intracellular development makes it difficult to study. The genus Chlamydia has a unique life cycle in the cells they infect. The cell phagocytizes elementary bodies, which differentiate to reticulate bodies. The reticulate bodies then divide in a vacuole, which progressively enlarges to form intracellular inclusions. The reticulate bodies condense to form new elementary bodies, and after 48 hours, the inclusion bursts, destroying the host cell and liberating the elementary bodies, which in turn go on to parasitize new cells. In practice, how do we determine the presence of chlamydia in patients with trachoma? There are different types of tests one can use. The question is, what is the infrastructure and what is the budget available for this kind of diagnosis? The first possibility is to do a gem sustain, but the efficacy of this is weak, not even half the efficacy of culture or immunofluorescence. Second choice being immunofluorescence. For this, you need slide, fixers, alcohol and acetone, extremely difficult to transport to the villages, as the bottled products are inflammable, especially in the heat. There remains the possibility of showing that the bacterium circulates, even if the germ studied is dead. And this is why we chose PCR. It is a genetic amplification. We didn't choose culture either, because with chlamydia, the specimens have to be kept in liquid nitrogen transport media, or at minus 80 degrees. This is unrealistic. Useful only for very few theoretical studies in conditions of wealth with freezers, liquid nitrogen and hospitals at hand. If we are in the field, in villages where prevalence studies are being conducted, we must limit ourselves to finding the bacterial genome, which we can do using classic PCR or a real-time PCR analysis. We chose the PCR in real time with a Pacman probe so as to always work within closed tubes. This way there is no risk of cross-contamination, no burst of electrophoresis, and no additional procedure to perform after amplification, since the instrument, the robot that does the amplification, detects the amplicants that were amplified and have produced a fluorescent signal within a closed tube. Over the years, the global cartography of trachoma established by the WHO has become more and more precise. The question was eventually raised about the trachoma situation in northern Cameroon. The first statistics concerning trachoma prevalence in Cameroon date back to 1955. A team of French physicians, led by Dr. Cousseran, organized the screening of inhabitants for ocular diseases in the region of the Logomé and Chari, north of the country. Out of 400,000 people examined, 1,600 cases of trachoma were found. In 1989, Dr. Yves Mage found a trachoma-related blindness rate of 4.6% in the ophthalmology clinics run by the NGO Ophtalmo Sans Frontières in the far north of Cameroon. If more than 10% of children 1 to 10 years old have follicular trachoma, the SAFE strategy is applied to all inhabitants of the region. The SAFE strategy combines surgical management of patients with entropion tracheasis, antibiotic treatment of the entire population, hygiene education in villages and schools, and improvement in standard of living, especially by improving access to water. In 2006, in the Kolofata Health District, the prevalence of follicular trachoma was shown by a study to be 21%. This high figure was confirmed by a second study, done in 2007, that found 24% of all children 1 to 10 years old were affected by follicular trachoma and 7.5% by inflammatory trachoma. It was therefore decided to apply the SAFE strategy. Antibiotic treatment of trachoma has made considerable progress in the past 20 years. The use of sulfonamides was abandoned because of frequent allergies and replaced by tetracycline eye ointment, which remains one of the possible treatments and a cheap one for trachoma. However, to be effective, the treatment with tetracycline eye ointment must continue for several weeks, which explains the poor compliance associated with its use. In 1980, the discovery of azithromycin and 10 years later, its use in the treatment of trachoma revolutionized the management of the illness. Derived from erythromycin, this antibiotic has the advantages of a good bioavailability when taken per OS, very good tissue diffusion and intracellular penetration, as well as a prolonged half-life. This medicine was proven to be effective against trachoma when taken by mouse once a year for 3 years. Beginning in 1999, through the intermediary of the NGO ITI, Pfizer Laboratories began providing this treatment for free to countries endemic for trachoma. To date, more than 200 million antibiotic treatments have been distributed by ITI in 18 countries. Azithromycin has therefore logically become the standard treatment recommended by the WHO. In the mid-1990s, there appeared the systemic form of azithromycin, a well-known antibiotic active against chlamydia, and this azithromycin really gave birth to great hope that trachoma might be eliminated from the face of the earth in a way that might have seemed easy at first. It is thanks to the boost generated by the arrival of azithromycin that an organization was set up, the Alliance for the Elimination of Trachoma, by the year 2020. At around the same time, or a little later, there arose an interest in developing a formulation that would be active in the form of drops rather than using an antibiotic given systemically. The development of these drops has a number of advantages, in particular that of giving an ocular treatment while at the same time doing some health education with the groups of people being treated. These drops also have the advantage of treating the disease where it develops, on the infected conjunctiva, in contrast to the oral form, which has above all a systemic action and thus the theoretical risk of creating new resistance when used for mass treatment. The French pharmaceutical company Thea was the first to develop and commercialize a topical form of azithromycin. There has been a long-standing relationship between the Chibret family and trachoma. This began in the 19th century with Paul Chibret, founder of the SFO, who was very interested in the disease, but it was my father especially, soon after the Second World War, with the arrival of antibiotics, invested a lot of effort in the disease. As a matter of fact, the only photo he had in his office was that of Dr. Antoine, a woman ophthalmologist who crossed the Sahara, working on trachoma in the oasis and particularly among the Tuareg. As for myself, as soon as I heard that the WHO was urging the pharmaceutical industry to develop a topical azithromycin, I seized the opportunity to become involved with the disease myself. I did not think it would be so long, nearly 10 years, because azithromycin is a particularly unstable compound. So at the last moment we were able to find an excipient, miliol, capable of producing a solution that was stable. And most importantly, stable in a warm environment, which is essential for Africa. This, then, is how the Azithr adventure began. In 2004, a study is undertaken in Guinea and in Pakistan. It proves the effectiveness of azithromycin eye drops against trachoma. 3 groups of children with active trachoma are treated either with a single dose of oral azithromycin or with azithromycin eye drops morning and evening for 2 or 3 days. The results show oral azithromycin and azithromycin eye drops to be of equal effectiveness against trachoma in children. Thea Laboratories having agreed to freely provide the doses of eye drops, an agreement is drawn between Ophtalmos Sans Frontières and the Cameroonian health authorities for the launching of a campaign to treat the entire population of the Kolofata district. Having proven the effectiveness of azithr drops against trachoma in Guinea-Conakry and in Pakistan, which paved the way for Thea Laboratories to obtain authorization to put it on the market, it was decided, together with the NGO OSF, to undertake a vast treatment campaign in the district of Kolofata. In this area, trachoma has a very high prevalence, over 30%, and it was decided to treat the entire population, that is to say, over 100,000 people, once a year for 3 years. This required quite hefty logistics, since in the first year of treatment we had to ship more than 700,000 unidoses from Paris to Yaoundé, with a view to treating a population of over 100,000 people. Patients suffering from entropion trichiasis are detected every year during the treatment campaign and referred to the Kolofata hospital to be operated on free of charge in the ophthalmology service supported by Ophtalmos Sans Frontières. The standard technique is a Trabot procedure, which consists in everting the superior palpebral tarsus with a set of sutures. After skin disinfection with betadine, and then the local anesthetic, the eyelid is everted over a plaque. The tarsoconjunctival incision is made 3 mm from the free edge, in association with 2 lateral incisions. The tarsus is carefully dissected with scissors or a blade. Then 1 vicryl or silk suture is inserted through each side of the incision, forming a U-shaped point. Once tied, the ciliary edge is flipped forward and the eyelashes straightened. Two other sutures are placed on either side of the first one. An application of tetracycline eye ointment terminates the operation. Azithromycin eye drops are prescribed for 3 days. The sutures will be removed after 15 days. It takes a few weeks for the eyelid to regain their aesthetic appearance. 542 patients were operated on in 2006, 355 in 2009. Third element of the SAFE strategy, educating people about facial and environmental hygiene. Since 2006, educational activities have been undertaken several times a year with the help of posters and health talks conducted in schools and villages. Mrs. Serpin, a teacher, initiated the educational work in the schools under the direction of Mr. Mayaga, Areas Inspector for Primary Schools. A health promoter, Mrs. Germaine Johnway, took over and continues to visit the various schools in the district. During these one-hour sessions, the children learn simple facts about the illness, its treatment, and the importance of good hygiene. Teachers are likewise encouraged to verify the cleanliness of their pupils' face before class, and if possible, to provide a supply of water and soap for washing. Teaching aids and comic books about Tracoma are distributed to them for use in class. The final part of the SAFE strategy, improving living conditions. This requires a more complex, multidisciplinary approach involving the political authorities and the village communities. 2 points must be considered. Access to water that facilitates corporal and environmental hygiene, as well as management of organic refuse that attracts flies, 2 factors in the transmission of trachoma. To resolve the problems of access to water implies constructing wells in villages that have none. This requires public and private funding, as well as a material and financial contribution from the inhabitants. However, such participation is not so easily achieved. In 2008 and 2009, 4 wells were built by the Cameroon government and 2 by the Association Friends of Kolofata. A project financed by the Peace Corps to construct 2 wells is in its early stages. In addition, it is not easy to change ancestral habits. The construction of proper latrines, simple measures of hygiene, better management of livestock in the compounds can only be imposed if existing systems are well understood and only after considerable time has been spent working with villagers. This last component of the SAFE strategy is therefore difficult to put into practice and will probably require many years of work in the field. After 2 years of the SAFE strategy, results in the Kolofata health districts are excellent. The prevalence of active trachoma has fallen by 80%, from 31% to 3.5%, below the threshold of 5%, which, according to the experts, signifies the interruption of trachoma transmission. The eye drop formulation of azithromycin has been formally proven to be effective in the mass treatment of active trachoma. Secondary effects were rare and minor. A final antibiotic distribution was undertaken in January 2010 and will be followed up in early 2011 by a new prevalence study. The fight against trachoma is thus well underway in Cameroon. The work accomplished in the Kolofata Health District has been fruitful and can now serve as a model for the rest of the country. The success of this action is directly linked to the organization of the project, the campaign, and the use of azithromycin eye drops, at present the antibiotic of choice for trachoma.